Okay, so first, I will try to show you the creation wizard using this example with the complete components, including cell, nucleus, and the vesicles in 3 channels. Okay, so for this single cell image, I think it should be okay to use parts or surface to easily create the number counting, do the number counting for this single cell. But when it comes to the multiple cell in the single field of view, then it will be very easy to use Imaris Cell to do this kind of job, to separate each cells. Here for the demonstration, the first demo, I will just use this one. So here to create a cell, just add new cells. So again, this creation wizard will pop up and you can follow the instructions here to go through the steps and get automatic results, detection result. So here we can select choose one of the detection type. In this example, I'm going to use the nuclear cell and the vesicles. So the first step is to detect the nuclei, which is actually using the surface algorithm. So we have to choose the channel for our nuclei and give the diameter. So I go to slice view and measure the size of the nucleus is about 15 micron. And we can expand the advanced options. So as you can see here, it's very similar to surface. So we can do the smoothing and we can have the background subtraction to have a better thresholding. And if we have multiple cells with nuclei standing and they are close together, so we can use the... Yeah, okay. There is some echo. So we can enable this split nuclei by C points, same as in the surface creation. Here I only have one nuclei, so I'm not going to enable this option. So here now we can see the thresholding of this surface area. Sometimes it will display as the slice view. If you want to see the volume view, you can change to settings, switch to settings and select the volume display. It depends on the graphic card. Sometimes this may not work. Okay, so now I can adjust the threshold to make sure the nuclei is covered by this threshold and go next, like this. Okay. So second step is to detect the cell. Now I can change to my green channel and go to settings, make my nucleus invisible so I can have a better view of this cell. So here I'm doing the cytoplasm staining, so I should select the detect cell boundary from cytoplasm and use green channel. Also, this is surface detection, so I can do the same thing. Have the same option here. So the sphere size. So here maybe I can give a larger range in order to remove some uneven staining in the middle of this cell. So maybe I give a larger number, about 30, and try like this. So whole cell body is covered. Sometimes due to the staining, you may see the hole in the middle. In this case, here I try to use the large number to smooth out these holes, to fill these holes. So it looks pretty okay here. So you can try to adjust these numbers to have a better thresholding for your images. And here again, we have the option to split touching cells. Then it can split by one nucleus per cell if you have nucleus staining, or if you don't have the nucleus staining, we can try to use this C points option as well. And then we have the options for nuclei. For example, if they're due to some reason, there are some nuclei outside the cells, we have the option to expand cells on nuclei or just erase nuclei outside the cell. So usually maybe use these options you can use. So here, this is my cell staining looks like. Cell segmentation. Go next. Third, last detection is about the vesicles. So here again, I try to disable my cells so I can view my vesicles better. Then in the vesicle detection, we have the options to add different detection and vesicle type. So if you have multiple vesicles you want to detect, you can add new type from here. So you will be able to select it from this drop-down list. So now I have only one, so I have the red channel, and this is about same as spot detection. So we can try to look for the size of these vesicles. It's about 1.3. So I'm going to use 1.3. And we can detect the different vesicle size as well. If you are interested in the size of these vesicles, you can detect different size, like in the spot creation. Okay, so next step, like this. Okay, go to next. So here, for the spot size, if you recall the previous sections, you have to make this thresholding area covers the size of your cover the area of your staining, and these two algorithms are how the diameter will be calculated. So I will try to use it like this. Okay, so now I have different size of these vesicles. Okay, so after all three components are created, now I can go to the setting tab and enable the display of cell and the nucleus. Then sometimes it will be easier for if you have a 2D image, maybe it's easier to just display in the slicer view. So it will show you a single Z section like this. But this is 3D image, so maybe I can try to use the volume display. And if you switch to the color tab, you will be able to use different colors for each component. So for cell, I want to change to best color and maybe give it transparent so I can see something inside. And for the nuclei, I can switch to blue as well, and also give some transparency. And the vesicle, I'm just stick to red, so like this. Now I can see my nucleus and my vesicles. And most important is here, the statistic. So of course you can see the number of cells. So maybe I have some mistake here. I leave some small nuclei here, so it is counted as the second cells. But in this case, I can go back to this detection and go to cells and try to give a better result from here. So I turn on. So 1 of 1 selected. Okay, so that should be okay. I need to check which one is detected. But that should be okay. So let me go back here. So cell gas again, try to make it transparent like this. Okay, so go to the detection and I can check which cell is detected here. So there looks like there is a small one here, which is a false detection. So I can, like in the previous step, I can delete, try to remove it from redo the creation, or I can just go here and maybe I have selected these small ones, which I want to delete. So I can go to the edit tab and delete it. Yeah, I think there is a false detection here. So anyway, here, I just want to show you the results. So as you can see in the statistic, you will be able to see these results categorized into cell, nucleus, and the vesicles. So in our statistic list, you can see the basic cell information about, including the area and position, cytoplasm, number of vesicles, etc. So this is easily categorized into each cell. So now, if you have multiple cells in the image, you will be able to click on each cell and get this result. For example, how many vesicles are inside the cytoplasm for this specific cells, and also cell number of nuclei, cytoplasm volume ratio, etc. For the nucleus, also we have the similar statistic. And for vesicles, we have the size, number of cells, vesicles. If there is some tracking, you have this tracking result as well. Position in the cytoplasm, etc. So you can see now if we have some questions related to each cell, for example, the volume ratio between the nucleus and the cell, then we can answer easily by using the cell modules here. Okay, and also we have the same filter tab we can use to filter out the different cells. And if you go to the edit tab, we can actually export these components we created from the cell module to surface or spots for further analysis purpose, or just for maybe a beautiful surface display. So for example, as you can see in the color tab here, we don't have as many materials as in surface for cells. So it looks maybe not as good as if we render it in the surface. So here, if I want to render this nucleus and the cell using the surface material, I can try to go here and export cell to surface. Also, I can export nuclei to surface. So now in my cell, I can just turn off the cell and the nucleus and just leave the vesicle. And here for my surface, cell surface, I can use this beautiful rendering mode to have a better display like this. Okay, so now this is one of the cells I have created, and I can take a snapshot or do the animation. In the meantime, I can still have this statistic. Okay, so that is one basic workflow to detect the different cell components using cell modules.